Good morning, and thank you all for tuning in to listen to some of my research related to identifying targetable pathways based on differential metabolic responses in drug metabolism in a breast cancer model. But first, I would like to thank the program committee for the invitation to share my work at this international forum. So to start with a bit of background for this project is that we know some breast cancer patients have poor treatment responses, which can result from a number of factors, and where I am most interested in race and ethnicity, genetic impairments associated with drug metabolism, and environmental causes specifically related to our diet. And because we also know that diets resulting in obesity, especially related to overconsumption of simple carbs plus saturated or trans fats, with low amounts of fiber and lean protein, characterizing westernized or the standard American dietary patterns, can also disrupt drug metabolism, specifically related to absorption or excretion, and cause diminished efficacy. Now, in some of my previous studies, I determined differences in metabolic and inflammatory signatures in 4 genetically distinct breast cancer cell lines representing hormone-responsive or triple-negative subtypes of disease. And today, I will present an expansion of that work, showing data for the cells alone and in response to the chemotherapeutic paclitaxel, or TAX as it will be abbreviated from now on. In another study, differential metabolic profiles were identified in tumors across all subtypes of disease in a cohort of African American women being treated at the Howard University Cancer Center in Washington, D.C., in collaboration with Dr. Jasmine Kanan and Robert Copeland. And then most recently, I demonstrated that a high-fat, high-carb diet resulted in faster growth and larger mammary tumors in a mouse model of basal-like breast cancer compared to a high-protein diet with more fiber content. And so again, because differences in treatment response can be based on ethnicity, other genetic factors, host metabolic health or immune system integrity, where chronic inflammation plays a major role, in the initial cell-based study, we used broad-spectrum NMR metabolomics and identified metabolic differences between these 4 cell lines, where the first 3 are derived from tumors of Caucasian women, compared to the 4th, which is derived from a tumor of an African American woman. And the MCF7 and BT474 cell lines represent hormone-responsive luminal A subtypes, compared to the 2 MDA lines, which are both triple negative. And several inflammatory cytokines were also correlated in this study to the literature based on clinical subtype aggressiveness and metastatic potential. And we found that these cytokines were specifically more elevated in that African American derived triple negative MDA 468 line. And again, this is in response to TAX treatment. And then finally, using an integrated pathway analysis approach, comparing differential metabolites and cytokines from just the 2 triple negative lines, we showed further distinction in treatment response mechanisms, including 2 with the potential for manipulation using nutrients related to carbohydrate or lipid metabolism. But I wanted to investigate the role of genetic polymorphisms, or SNPs, specifically in genes involved with taxane metabolism, as a further expansion to this study. So to again find nutritionally relevant gene metabolite interactions that can be targeted by a nutrient modification approach to ultimately improve treatment response. And so with an addition of 4 more cell lines, all listed now in this table here, and also including some of the patient demographics and clinical or pathological tumor characteristics, I set out to accomplish these 3 goals. After treating the cells with tax at a uniform LD50 across the 4 new lines of 100 nanograms per mil and collected samples, but this time used a high-resolution untargeted LC-MS-based metabolomics approach for data analysis. Then I also started with this panel of candidate genes for the initial polymorphism screen, which can be better visualized from this taxane pathway figure previously published by Oshiro and colleagues. And here, as you can see on the pathway map or on the list, it includes genes responsible for either direct phase 1 metabolism, regulation of CYPs, transporters, glutathione detoxification or clearance genes, as well as a few others. And again, using multivariate analyses for visualization, we can see that both in the unsupervised principal component analysis or the supervised orthogonal partial least squares discriminant analysis, that we have demonstrated a strong metabolic distinction between the 4 new cell lines here. And also, these plots show that there are differences based on both subtype representation and ethnicity derivation, where here the black dots represent the HCC 1937 triple negative line derived from a Caucasian woman's tumor, compared to the blue dots, or the HCC 1806 line, which are also triple negative, but derived from a tumor of an African American woman. And clearly they're very different, being separated by both the primary and secondary axes in the multivariate space. Now, comparing the other 2 African American derived cell lines here in green and red, which also are presumably different metabolically based on also another subtype difference, but this time either ER positive status versus HER2 enrichment. But overall, the patterns of separation in either suggest that the cell type itself is a stronger clinical or metabolic differentiator than treatment status, as we see the cells treated or not cluster still together. And this is a phenomenon we also saw in the first 4 cell lines in the initial study. So of course, this begs the question of what is it then about the unique cell line specific qualities that's really driving treatment response differences, and how could this translate to what we see in patient populations? So now let's go through what some of those specific metabolites are that characterize the individual cell line treatment responses to tax. And they're shown here for the first 4 cell lines in these tables. And I'll just highlight the fact, or point out the fact, that the metabolites in red represent unique response-driven markers for the media controls versus the tax-treated cells in each cell line. And then for the second set of 4 cell lines, the HCC lines, again, we can see that the HCC 1806 triple negative line showed the most robust numbers of unique markers, with about 5 or 6 total. And unfortunately, even after library matching to our in-house library or some external databases, there were no confident matches for metabolite annotations in this cell line. And next I show some selected pathway analysis results using the software platform Jingo. And here we see the significantly perturbed pathways differentiating, again, each individual line based on its own response to tax treatment. And here the selection of what you see is simply based on space constraints, not allowing me to show the full list under each line. But I would like to point out that I've highlighted, again, some of the uniquely perturbed pathways for each individual cell line by these green stars. And for 5 of the lines shown, they each represent differences across the different clinical subtypes of disease. And finally, in another GeneGo analysis, this time using the multivariate determined differential metabolites between the 4 African American derived and the 4 Caucasian derived cell lines, we found 31 endogenous pathways that were significantly perturbed between these cell types by ethnicity. And here we show that some are more prominent based just on the cell types themselves, indicated by the orange bars in the graph, compared to being more prominent based on treatment response differences, and those are shown by the blue bars. And namely, we found that this pathway related to vitamin, mediator, and cofactor metabolism, specific to biosynthesis transport, was very important, because when we go back to the context of our SNP results, where here I show 4 that were listed for either a transporter, a phase 1 drug metabolism CYP, and 2 glutathione detoxification genes, we know that they, which play roles in drug clearance. We can see here in the bottom table that 3 of the 4 show differential SNP genotypes. And then if we look back to the taxane metabolism figure here, in correlation to these data, we can see, excuse me, that we have 2 genes with polymorphisms that are directly reflected on the pathway. And here in the table, I've now also added the clinical subtypes, which allows us to see further genetic heterogeneity across the cell lines, and then based on the differential SNP profiles. Now finally, using another integrative analysis, this time between the significant ethnicity differentiated metabolites that were used for the previous GeneGo analysis, but this time also adding in the information based on the 4 candidate genes, 3 of which again showed polymorphisms, we kind of answered our million-dollar question of whether this approach could identify potentially targetable gene metabolite interactions, where here we show that some interactions occurred only between the untreated cells, but also when we looked in the context of tax treatment, we see that the lists are fairly similar, but the important point is there's a loss of interaction in this mechanism of interconnectivity between folic acid and fluorouracil pathways demonstrated in gastric cancer previously, and now we've demonstrated here in breast cancer model. But then I also wanted to point out this NRF2 antioxidant pathway association, in either case, treated or non, where in the database it was demonstrated in a smoking study using airway epithelial cells. But this is an important pathway because NRF2 is a nuclear transcription factor that regulates genes containing antioxidant response elements in their promoter regions. And many of these regulated genes encode proteins involved in injury response and inflammation, including the production of free radicals, and thus indicate oxidative stress. And so while it is widely implicated in cancer for increasing the antioxidant and detoxification capability of cancer cells, inhibiting it still represents a good targetable strategy. And that's evidenced by just a quick literature search that I did, and I listed 7 interesting publications, and some of which you'll see actually use nutritional modification or interventions to show and demonstrated improvement in chemotherapeutic response directly in relation to this pathway. So again, this was a reassuring discovery for us, and we feel like we are on the right track for finding ways to use nutritional approaches to ultimately improve treatment outcomes for patients with breast cancer. And with that, I really hope you have enjoyed the work I've shared. And again, many thanks to the organizing committee for the invitation. So finally, I would like to acknowledge and give many thanks to all my exceptional colleagues who collaborated with me to do this work, as well as the institutional funding provided by the Nutrition Research Institute in Kannapolis, North Carolina. Thank you for listening.